All right. Good afternoon, everyone. Today's webcast is being recorded and will be posted on the ELOSC website. I would like to welcome everyone to today's webcast of the Eastern Lake Ontario Swallow-wort Collaborative. My name is Robert Smith. I'm the Terrestrial Restoration and Resiliency Coordinator for the SLELO-PRISM and the Nature Conservancy in New York, and the host of today's webcast. I would like to ask participants to mute your audio to reduce feedback during today's webcast. Our webcast this afternoon is designed to continue our discussion on managing swallow-wort, one of the Northeast's more aggressive invasive species. Today's webcast will include presentations from Carrie Brown-Lima and Audrey Bowe from the New York Invasive Species Research Institute, and Rob Williams from the St. Lawrence Eastern Lake Ontario Partnership for Regional Invasive Species Management. There will be opportunity for questions after the presentation. The Eastern Lake Ontario Swallow-wort Collaborative, or ELOSC, is a platform for sharing the most up-to-date research and best management practices to inform stakeholders about the prevention and engagement of our management of this highly aggressive invasive species. We hope the ELOSC will foster greater cooperation and collaboration between different stakeholder groups to improve control and spread prevention focused solely on swallow-wort. Additionally, this collaborative helps to transfer knowledge between researchers and managers, share advances in science and management, provide technical assistance, and connect the stakeholder community. Our tagline for this collaborative is linking people, information, and action through enhanced communication. Cornell Collaborative includes current collaborators include Cornell Cooperative Extension, the Nature Conservancy, the New York Invasive Species Research Institute, the University of Rhode Island, New York State Department of Parks, Recreation, and Historic Preservation, Wells College, and the 1,000 Islands Land Trust. Any stakeholder organization that embraces this effort is invited to become a collaborator. It is quite informal, no papers to sign, just a willingness to share information. Please contact me at robert.l.smith@tnc.org or any one of our collaborators should you desire to collaborate. I would now like to introduce our guest speakers. Carrie Brown-Lima, Audrey Bowe, and Rob Williams. Carrie and Audrey will be our first presenters and are both from the New York Invasive Species Research Institute. Carrie as the director and Audrey as the project coordinator. This institute works closely with research scientists, state and federal agencies, the New York Invasive Species Council, and advisory committee and regional managers and stakeholders to promote innovation and improve the scientific basis of invasive species management. Rob Williams will be our second and last presenter for the day. He is the Invasive Species Program Manager for the St. Lawrence Eastern Lake Ontario Partnership for Regional Invasive Species Management, or SLELO-PRISM. SLELO-PRISM's mission is to protect native habitats, biodiversity, natural areas, and freshwater resources by using a collaborative and integrated approach to invasive species management with emphasis on prevention, early detection, rapid response, education, and outreach. Please share your questions in the chat box and we will get to them after the presentation. Carrie, Audrey, the floor is yours.
All right. Can you see my screen, Robert?
Yes.
Okay. There we go. All right, hello everyone, good afternoon. Thanks so much, Robert and Rob, for the invitation to come and share a little bit of the work we've been doing at the Research Institute and with all of our many collaborators. The title of our talk, From Research to Implementation, an overview of swallow-wort biocontrol release projects in New York State. And as Robert mentioned, I'm Carrie Brown-Lima and I direct the Research Institute. I'm going to start just by giving a little overview and background on how we became involved in this work. And then I'm going to pass the screen to the project coordinator, Audrey Bowe, who will tell a little more about what we did this summer and what we've been doing for the last couple of years. So I'd like to just first of all take the opportunity to share a little bit about the Research Institute. It was established in 2008, and our mission is to coordinate invasive species research to help prevent and manage the impact of invasive species in New York and beyond. So we work at the interface of research and of management and policy decisions. And so we coordinate research to meet the needs of the folks out in the field who need it, and we also communicate their needs to the research community. So, for example, every year we ask our stakeholders in New York State what their research needs are, and then, like I just said, then we'll communicate those and make sure that information gets out to the researchers, and that we also work to advance those projects. And we also foster knowledge exchange. Many of you have probably participated in our conferences or webinars. We also serve on many committees and boards and are involved in different networks, so we can make sure that we are keeping people engaged and being at the forefront of what's happening in invasive species work. And then we also support managers. Oh, there we go. And researchers, we synthesize research, we communicate and seek funding opportunities, and so on. And so we're really just trying to make sure that those lines of communication are really strong between these two worlds. So how we became involved in the swallow-wort work is every year when we put out this survey, for a couple of years in a row, advancing swallow-wort biocontrol kept coming back as a top research need, with folks saying they really wanted to see that advance. And so with that in mind, we started identifying different collaborators at different universities, and started really trying to draft up what would need to happen to make sure we could implement biocontrol for swallow-wort in New York State. And then we sought funding for both the research and extension and outreach on this topic. So as many of you know, the research, the biocontrol agent that we've been focusing on is a defoliating moth, Hypena opulenta. It comes from Ukraine forests originally. It generates 2 generations per summer, and Canada is much quicker at their approval process. They released it back in 2003. However, here in the United States, we only received approval in 2018. So the work that we're talking about with the releases really is starting in 2018. And so, as I mentioned, this process is kind of long in the United States and bureaucratic, but the objective is to make sure that these agents, the biocontrol agents, are actually very well screened and have been thoroughly evaluated before they are allowed to be released. And so, as you can see from these timelines, it can take anywhere from 5 to 15 years to get a biocontrol agent approved. However, and I'm not going to go deep into this process today, but what I did want to point out is that the process doesn't end once an agent is approved. Once the agent is approved, then we have to actually think about how are we going to get this agent out in the field and distribute it to the different locations where the target invasive species is located. And we also need to develop a trial strategy. So we need to see how best to release them, how to collect data, how to monitor them, what are the best ways to produce them in large quantities, so mass production and distribution. And then we also need to get that information out to the people who are going to be doing the releases and the monitoring in the long run. And then we also need to have an idea in place, a system in place for how we're going to evaluate whether our project was successful or not. So what is successful biocontrol agent? Well, for it to be approved to begin with, as I mentioned, it can't eat anything but its target species. So it has to go through this rigorous thing to make sure that it's not going to start eating everything else and that it's only going to eat the target species. And then it also needs to not only eat it, but it also needs to feed enough on it so that it can actually reduce its population. But for a program to be deemed successful, which is what we're trying to test right now with Swallow-wort, whether it's successful or not, first of all, it has to be able to survive once it gets released out into the wild in its new home. So that's establishment of the agent in the introduced range. And then once it's established, we actually want to see it decrease the population. So in this case, we want to see the Swallow-wort populations go down. And then we want to see the native species and ecosystems that we care about and the ones that we're looking to protect rebound. And so just a little diagram here to say, you know, this ideally the weed density is high when you release a biocontrol agent, and then its population builds up and starts reducing the weed population, the target invasive species population. And then as that population goes down, also the biocontrols agent's population will go down along with it because it can only feed on that species. And that will hopefully allow for the desirable vegetation to increase. And so here in New York, in looking at this next step of what happens after a biocontrol agent is approved, here at the Research Institute, we partnered with a bunch of partners that Audrey is going to talk about in her portion of this presentation for a two-tiered program to advance biocontrol of Swallow-wort. So the first aspect is research. Audrey will be telling you about the 5 different universities that we've been working with and how the Department of Transportation provided funding for us to conduct Swallow-wort experimental mass rearing and releases and develop monitoring protocols in New York. And then we also received funding from the USDA Smith-Lever grant to partner with Cornell Cooperative Extension to develop the outreach and implementation program so we can get the information out to land managers about this biocontrol agent in general, and also to develop the protocols and simplify the protocols, provide trainings and outreach materials so that it'll be easier for land managers to start actually doing these releases on their own and still collect the valuable data that we need to evaluate its success. So with that in mind, I'd like to pass the screen to Audrey, who can then tell you more about these 2 tiers of our program and some of the work that they've done this summer.
Great, thank you, Carrie. Can you hear me and can you see my screen?
Yes, but you're not in presentation mode.
How's that?
Better.
Okay, great. Yeah, thank you so much, Carrie, and thank you to Rob for inviting us to give this presentation today. As Carrie mentioned, we're at the Research Institute. We're currently working with 2 groups focused on Swallow-wort biocontrol, the first of which is the research group, and then the outreach group. And so I just wanted to give a brief overview of the research group and sort of an update on what they've been working on lately, and then really dive into the work that we've been able to do this past summer with our outreach group. And so you're seeing on your screen the members of our Swallow-wort research group, which includes Dr. Andrea Davalos at SUNY Cortland, Dr. Jackie Schnurr at Wells, and both of them study plant ecology, invasion ecology, and then Drs. Dylan Perry and Lindsay Milbrath at ESF and the USDA, respectively, and they're on the entomology side, so dealing with rearing and establishment of the Swallow-wort agent, Hypena. And then Carrie and I are sort of in this intermediate bridging role at the Research Institute, where we're interacting with this group to understand what they're doing and try and bring that information back to the outreach group and disseminate it more widely. And as Carrie mentioned, this group is funded thanks to the Department of Transportation. And so the overriding objectives of the research group, this group came together around the time that Hypena was approved for release in the United States in 2017. And with that in mind, they're interested in questions of both implementation. So how can we develop the optimal mass rearing techniques that you need to get the number of agents out into the field that you would want, as well as understanding how well does this agent establish in the field in the United States, and what are the factors that sort of influence that. And then on the vegetation side of things, as Carrie mentioned, a biocontrol agent isn't truly successful unless you're having that impact on the target plant. And so understanding and being able to quantify the impact of Hypena opulenta on swallow-wort in the field under different conditions, as well as whether or not we're seeing an improvement in conditions for native plants. And so based on these questions, the research group has established 10 field sites where they're running experiments in central New York, doing vegetation monitoring to capture sort of what's happening in the time before agents are released, to get sort of a baseline set of data, as well as a seed emergence study to understand how swallow-wort is recruiting. And this, importantly and excitingly, was the first year that agents were released at these sites. So they were released into these insect cages, basically 6 by 6 by 6 walk-ins covered in mesh, to establish the agents there. And so Dylan Perry, who's pictured here in the top left, has a colony that he's rearing at ESF, and he was able to release 30 males and 30 female adults in each of these 10 cages in June of this year. And revisiting these cages, they were able to see about 100% defoliation in 6 out of 10 of the cages, almost complete defoliation in 2 more, and then modest defoliation at the remaining 2. So overall, great initial establishment at these sites. And he also conducted some larval releases supplementally at these sites. Between 200 and 500 larvae were added. And he also noted that he had seen movement of agents out of the cages between 40 and 60 feet away. So that was exciting dispersal. And so that's what's been going on for the research group this past summer. And they intend to continue monitoring the moth establishment and noting the defoliation at these sites, as well as engage in some more studies trying to understand how the pupa, which is the overwintering stage of this agent, is going to do in the field. And so looking at predation and possible mortality over the winter, as well as continued vegetation monitoring. So now that these agents have been released, all the vegetation monitoring in these sites is going to be post-release information. So we'll start to see if swallow-wort is being impacted. And so that was a little bit about the work that the research group is doing. Now I want to talk about the work that we've been doing with the outreach group. And so this is the core team depicted on the screen here of our members. And so we've got expertise in extension and developing outreach materials and trainings in the form of Sharon Bachman, who is based in Erie County at Cornell Cooperative Extension, and then Arlene Wilson, Maggie Marr, and Laura Bailey, who are all out of CCE Yates. And then Carrie and I, who have been working with them and with the research group to sort of bring together information and expertise around this topic. Oops. Oh. And this is, I should note, supported by a Smith Lever grant. And so as Carrie mentioned, bringing a biocontrol into implementation involves a lot of transfer of knowledge between research and researchers and those who are managing in the field. And so with CCE, we're trying to create both awareness, outreach materials, and also develop some tools to help people who will eventually, hopefully, be using these biocontrols. And so these are our objectives here, which includes increasing awareness, developing a monitoring protocol, establishing demonstration cages, and developing training materials, and a database where we can archive all the information that's hopefully being collected through monitoring. And so the bulk of the work that we did this past summer fell under these two objectives. So developing that standardized monitoring protocol that's accessible for many people to use, and also establishing the sites themselves. And so to develop this monitoring protocol, the research group was already actively monitoring vegetation. And so we worked with them. We went out in the field to see how they had set up their field sites, to identify the conditions that we would need to release biocontrols under, and also the vegetation attributes that we would want to collect to be able to say something about agent impacts. And so this is the schematic that we ended up using based on sort of reducing the scheme that the Swallow-wort research group had developed. So we ended up using a series of 10 meter square quadrats where we're monitoring vegetation within these, so swallow-wort and whatever else is growing with them, and also establishing these temporary insect release cages that are the same cages that the research group I mentioned that the research group was using earlier. And the reason for using these is because when you go to assess establishment of Hypena, it has a reputation for dispersing pretty widely. And so if you were to conduct a release in an open release, the likelihood that you would be able to effectively see, hey, did my agent survive? Are they reproducing? Is really, really limited. But this way, because everybody's contained in the same area, you both increase the likelihood that your moths will find each other, and also that you'll find your moths. So you'll be able to relocate them and truly understand what's going on. And so these are temporary, and they're really just to get things going in the field. And so I mentioned we worked with the Swallow-wort outreach group to develop the vegetation monitoring protocol. And so basically the information that we ultimately pared down their much larger data collection scheme to fell under two categories. One of percent cover, which will allow us to see with a broad brush changes in swallow-wort percentage, and also subsequent change with other vegetation that's surrounding it. And so the hope is that you would see swallow-wort percent cover decrease over time and native cover increase. But we also put a category in here for documenting whether or not other invasive species were present. It's definitely been the case in the past in some environments where you can introduce or manage a biocontrol for or manage one invasive plant and then be successful in that, but have it just be replaced by another invasive species. And so we wanted to create a space to capture that, as well as capture changes in native vegetation. And then we're also collecting in those meter squared monitoring locations, swallow-wort density at different life stages. So understanding how many seedlings there are, how many sort of immature stems, so non-reproductive, non-flowering within that area, and how many flowering individuals to sort of get a sense for population structure over time to see how that's changing. And so as far as the insect monitoring, within those cages that we were planning to release in, we were lucky that Lindsay Milbrath from the USDA had already developed something to monitor in those cages that we were just able to adopt. And so we plan to document presence or absence when you went to visit. Did you see the larva? Did you see the pupa? Did you see moths? Did you see eggs? Yes or no? And then also observe defoliation. So did you see evidence of feeding, of insects doing their thing? So after developing those protocols and establishing sort of what we wanted field sites to look like, we were able to work with our partners, and our collaborators worked with their partners to establish release locations. And so we ended up this summer having 4 release sites. The furthest on the left is near Buffalo. It's working with land owned by the town of Clarence. And this was with our collaborator in Erie, who was monitoring. This second to the left site marks Ganondigan State Historic Site. And so working with collaborators there, with the environmental field team and New York State Parks helping us monitor. And then 2 other sites that we partnered with the Thousand Islands Land Trust and also SLELO to establish locations up there with their partners. And we also are grateful to private landowners in the Thousand Islands who allowed us to set up sites on their land. And so this is what the setup of the vegetation monitoring looked like in the field. This was at Ganondigan. And you can see these are in the picture on the left, you can see the cage going up in the background, the insect cage, and then we established the permanent monitoring locations for the vegetation and collected all of that data. And this was in late June of this summer. And then after we had all of those, all of the pre-release vegetation data and the insect cages set up, the next step was to conduct releases of the biocontrol agents. And so based on conversations that we had had with the research group, we initially planned to release adult moths. There are sort of pros and cons to releasing adult moths versus larvae versus pupa. And it had sounded like it could be most promising to release adult moths. You would be able to be more certain that they would survive and that they would potentially lay eggs and lay the groundwork for the next generation that would eat your swallow-wort in your cage. However, unfortunately, due to some challenges posed by COVID this year, as well as some unexpected transition into diapause, so an overwintering stage of some of the agents in Dillon's colony, where we were hoping to source our moths. We were not able to get adults. However, we were rescued by Lisa Tewksbury, who has a lab at the University of Rhode Island, who was able to send us some pupa. And so at each site, we ended up releasing in these cages about 14 males and about 26 female pupa. And they went into these pupal emergence containers that we hung from the ceilings of these cages. And so the idea was that they could hatch. They were in a vermiculite substrate, and when they hatched, they could just fly out the door and establish. We also had people changing and maintaining a jar of honey water as a potential supplemental food resource for any adult moths hanging in the corner. And this is on the right, just an image of someone hanging up that honey water. And so these releases, we received the pupa on the 21st of July and made all of the releases on the 22nd. And the reason we hung this this device up was to keep it out of reach of potential predators. Apparently mice and other rodents will really decimate your pupa if you let them. And so here are some more photos of releases going on in the field. So hanging up the chamber, what the completed cage looked like, as well as hanging the honey water. And so once Carrie and I had sort of distributed the agents, our partners were the ones who were doing the sort of test run of the monitoring protocol and keeping track of what was going on insect-wise in these cages. So they were going out once a week to check in on things, to see if they saw any of the agents, if they saw any evidence. And each field site had sort of a different experience. So I'm going to run through each of the sites and show you what everyone found. So the first site I'm going to talk about is Grenadier Island, which is actually up in the Thousand Islands. This was monitored by a private landowner with support from Rob Smith of the SLELO-PRISM and Brandon Hollis of the Thousand Islands Land Trust. And so this was obviously on an island in an open field. You can see on the right that was predominantly swallow-wort. And so you can see the July 22nd release happening. And then on July 30th, they had 4 moths that were observed in the cage. However, after those 4 moths that were observed, there was never any larval establishment in this cage from that time. And so we're fairly confident that this cage did not have the agents established. The next release I'm going to talk about is the one on Carlton Island, also up in the Thousand Islands, and monitored by private landowners. And so this site was more forested. You can see the cage on the right depicted here. And again, released on July 22nd. And unfortunately for this cage, none of the pupa ever emerged. So there were no moths that were found in this cage and no establishment. And this could have been for a variety of reasons. We're really not entirely sure. We suspect that this was a very hot period, and this is a plastic container, and so it's quite possible that it acted as a little greenhouse and got a little bit too hot for these pupa, too hot too fast, and they never got the chance to emerge, or it could have had to do with pupal quality. This is our Ganondigan State Historic Site down in central New York, and this was monitored by Laura Bailey out of CCE Yates, one of our collaborators. And the site at Ganondigan is an open field. And so here's our release on the 22nd. And then on August 12th, a few moths were observed and some feeding damage, which may or may not have been Hypena. However, ultimately, no larvae were seen in this cage. And so that establishment was also probably a failure. And last but not least, we have our Town of Clarence site near Buffalo, which was monitored by Sharon Bachman out of CCE Erie. And this is a forest site located in this really cool open oak forest habitat on the Niagara Escarpment. And so in the bottom left, you can see the release. And then on August 5th, there were observations of several moths. And by August 19th, a little bit of feeding damage as well as some tiny larvae were observed. And then by September 22nd, Sharon was observing significant feeding damage and larger larval instars. And so we consider this, thankfully, hopefully a successful establishment. We'll see. We hope that larvae at this stage will either already or soon be pupa, and that these will survive the winter, but to be determined. And so we had really variable experiences and variable success with establishment of these agents in our cages, which could have been due to a variety of factors, including heat and pupal deaths. However, we plan to continue with 2021 vegetation monitoring at our permanent locations and also make additional releases, hopefully that will be adults into our cages. And so based on what we've learned and how we've developed the protocols, we're working with our CCE partners on developing training materials for those who might want to conduct or who will be conducting releases in the future. And so this includes short videos on how to monitor inside a cage, how to change the honey water, as well as a pocket guide and other outreach material for information on Hypena and monitoring in general. And we're also hoping to develop a form working with IMAP Invasive to collect the vegetation and insect release data. So there's an archive for all of that, and that's an easy way to enter it for those working in the field. So concerns and challenges that we've experienced with this program, a big one for us has been agent availability. So Hypena does not appear to be the easiest agent to maintain in a lab colony and rear. At least it's relatively unpredictable in certain ways. We experienced difficulty in both 2019 and 2020. In 2019, a lot of colonies, lab-raised colonies, experienced a high level of a pathogen called microsporidia, which really led to low colony numbers and limited availability of agents. And then this past year, we were also limited in our ability to get adults. And so we're hopeful that this will get easier and improve over time as people and labs are better able to rear this agent. And another thing that's yet to be determined is overwintering success in our areas, in the research sites, as well as the outreach sites. And so trying to understand, is this going to be a big bottleneck? Will you lose a lot of your insects over the winter, or will they be successful? And going along with that, trying to understand, there's some question, it remains to be seen how well Hypena opulenta will establish in our region. And so that's an ongoing topic of research, and we'll see in the next few years, hopefully. A lot of agents and sometimes biocontrols in general take some time to get going and to build up population numbers. And so it can be, it could be a number of years before we see significant impacts to swallow-wort populations. And that's another thing, we might never have Hypena at high enough populations to control swallow-wort populations. And so that remains to be seen. And so moving forward, there are other swallow-wort biocontrol agents that are being developed. One in particular is a root feeder. And so potentially down the line, if that is approved or if that does move forward, being able to incorporate that into the program and into the monitoring. And we also hope to use this approach that we've developed at the Research Institute as sort of a template or a model for monitoring establishment and impact of future biocontrol agents for other invasive species. So in New York, there are a number of biocontrols relevant to invasive species that could be made available in the coming years. And so being able to adapt this sort of approach to be able to capture information on releases, as well as be able to monitor what's happening with those releases if they're having the impacts that we want them to. And so with that, I just want to say thank you to all of our partners and landowners who we've been able to and fortunate to work with. And thank you to everyone for listening today. I'm happy to take any questions. And I know Rob Williams has an update from Wehle State Park and the releases that they made there. Thank you.
Okay. Yeah, thank you, Audrey and Carrie, for sharing this information with our participants. We'll now move on to our next presenter, Rob Williams. So Rob, the floor is yours.
Thank you, Robert. Good afternoon, everyone. Bringing up my slides here. Hopefully everyone is seeing my cover slide at this point. So thanks for the opportunity to share this information. I think I'm going to take just a slightly different focus than what's been presented so far. So we've been working with our program here with the SLELO-PRISM and the Nature Conservancy with pretty much the same partners that Carrie and Audrey have been working with. And for the purpose of this presentation, we're all familiar with Swallow-wort. As you've heard, we have been working on 3 different sites up near the Thousand Islands. The first site, Carlton Island, and the second site, Grenadier Island. You've already heard from Audrey on those particular projects. And for the purpose of this presentation, I'm going to focus on the Wehle Forest down here and what we saw over this past summer with that particular site. And I just want to point out that Lindsay Milbrath is the primary investigator at this particular site. So the rest of what I'm going to share with you has to focus on the Wehle Forest. So similar to other release programs, the release process is delicate. Ideally, we want no or little predation on the larvae. We want to see adult emergence. We want to see eggs being laid. We want to see larval development. And as was previously mentioned, sometimes the timing of this can be tricky in terms of having the larvae go into diapause or achieving adult emergence. So this particular photo here in the upper left, you'll see the larvae being readied for release. For the Wehle site, they were placed in a bucket, if you will, which was designed so that the adults could emerge out the top of the bucket, which has a cover, and it helps protect the larvae and the adults from predation while they're in that bucket in the lower left. And then in the upper right, you'll see Lindsay Milbrath placing one of the buckets with the pupae in vermiculite in the buckets. Research cages, same size, 6 by 6 by 6. And this year, I'm happy to say that we had many egg-laying adult moths emerge. And although I've shown this as success, there's a lot more to it than just having adults emerge. You'll see in the Wehle site, which we had 2 cages, adults did emerge. You'll see a couple of adults here approaching the 10% honey water, which we had 2 jars in each cage. And we had, as you can see here, the larvae, which feed exclusively on swallow-wort, causing substantial defoliation. And I think the idea here is to achieve enough defoliation where the plants can't successfully photosynthesize and therefore they can't mature and produce seeds. So here you can see one of the instars doing its thing on the swallow-wort leaves. Repeat slide here. This is just an example of what they can do to the swallow-wort leaves in the stem. One of the things that we've noticed in the past is that you really only need about 50% defoliation to achieve stem mortality. So some of the science, again, this is focusing on the Wehle site. We had an initial release, and week 1, a week after our initial release and going into week 2, we had successful adult emergence. Beginning week 2 and into week 3 and 4, we had substantial larvae development, and it often was the case where they were too numerous to count. I think Robert went out there once and had caterpillars on his sleeve and on his shoes as well. And by week 4, we had over 90% defoliation of swallow-wort plants in the cage. So for us at this site to go from an initial release through successful adult emergence, misspelled there, apologize for that, substantial larvae development and over 90% defoliation in 4 weeks. We're looking at as a successful test release in the release cages. So here's a photograph of the before and after. The upper left is obviously one of the cages before the introduction of the Hypena. And then 4 weeks later, we had nearly complete defoliation within that cage. And once we had achieved or reached that 4-week, 5-week mark, the cages were removed and the insects and any larvae that may have been in there and any eggs were liberated into the wild. So it's hard enough to get adult emergence. So we did. We consider that successful. We had egg laying occur. We had the larval development followed by defoliation. So in terms of an initial in-cage test release, we're looking at this as fairly successful. Moving forward, cages have been removed. Adults, larvae, and eggs have been liberated into the wild. We believe that Hypena can overwinter in our climate. As was mentioned earlier, we don't know what the level of success will be on overwintering. We can hope for nominal predation during diapause, and then we want to conduct additional releases and allow the population to hopefully establish itself and continue to defoliate and suppress swallow-wort plants. A lot of those are unanswered questions, but that's what we're hoping to achieve. So in a nutshell, we had pretty good success at the Wehle site, and I tell you, it's really exciting when you go out to one of those cages and you see a whole bunch of adults flying around, and then you find larvae on the swallow-wort plants. It's a pretty good feeling. So that's all I have, and we have more information that'll be posted to the Swallow-wort Collaborative.org website. Thank you.
All right. Thank you, Rob, for sharing the information with our participants. Now I'd like to take some time to read questions from the chat box and take some audio questions if anybody wants to do it that way. We got a few in the chat box. Let me see what we got.
Robert, do you need me to stop sharing my screen?
Yeah, then I'll just bring up mine. All right, there we go. As soon as I can get my chat box back up.
Sure. So Robert, there is a question in here from Christine about why the Wehle release was could be viewed as more successful than some of the other releases, and what are the key differences. And I don't think I'm the best one to answer that, other than to say that I believe we received larvae at different stages from different sources. But I would actually reach out to Audrey or Carrie to see if they can shed more light on why we would achieve different levels of success or adult emergence. Carrie or Audrey, can you help?
Sure. Yeah, I can speak to that a little bit. So yes, like Rob said, we had a different source. So Rob's release was provided by Lindsay Milbrath, who's based at Cornell, and we got our agents And so he has his own separate colony that he was sourcing them from. And those insects were a different generation, so to speak, than the ones which we used, which had just been brought out of essentially their cold period, so their diapause. And so it's possible, I'm told that it's agents that have been sort of in diapause for longer can sometimes be less robust. And so I think that was a contributing factor. And we also were a little bit limited in terms of the number that we were able to release, just because that's what was available. And so I think that in some of these cages where we had moths emerge and seem to have survived, we might have had a mixed or like a very skewed gender ratio where, you know, maybe because we released more female pupa, they were all females and they never were able to mate. Yeah, I think just releasing a lower number overall probably made the cage more sensitive to predation. So losing one or two was you know, more detrimental to the cages in Grenadier and Carlton than the ones in Wehle, because Wehle had many more larvae that they were releasing. I forget, maybe Rob knows the number that were in each of the cages released, but we had a total of maybe 30 or 40 going into each.
The other thing, this is Carrie, that I wanted to mention, the difference between our sites and Rob's is that ours actually had to be shipped as well. And so just one extra stress on them. And it was also quite hot when we did our releases, so that could have impacted it as well.
All right, we have another question by Tom Wild. Says, do you have an expectation of how well a successful population will expand into the area? For example, into Chameau and other surrounding areas?
Yeah, tough question to answer. You know, we don't want to get our expectations real high on this. And that's part of the whole research phase, is to monitor the release sites to see how, you know, what the characteristics are of moving away from the initial release sites. So it's hard to create an expectation before you've done the research to determine how these things act out in the open.
I know in Canada, I think I see Rob Boucher's on the line, and he's been conducting releases in Canada for a while now. And I thought, I recall him saying they were found several miles from the original release site, but I don't know how successful those populations were at establishing. Rob Boucher, if you're on the line and want to comment.
Okay, let's see, we got another question from Robin. Says, do you think Hypena need a critical density of pale swallow-wort to maintain their population?
Sorry, can you hear me?
Yeah.
Yes.
Sorry, this is Rob Boucher from Canada. I was on mute. So,
Oh, okay, go ahead.
in answer to Carrie's question or comment, the moving up to about 2 kilometers within the first couple of years. So in terms of finding defoliation, that distance away, probably within 2 to 3 years of the initial releases. But what we're seeing now is I think they're spreading out into the sea of DSV that's out there, swallow-wort that's out there, and harder to detect in terms of the amount of area that has to be surveyed. So they're also selecting the darkest spots they can find. So if you're looking for them, it's best to look for them in understory forest, hedgerows, that sort of thing is where we're finding the largest amount of defoliation.
Okay, all right. Hey, thank you for helping with that question. So going back to Robin's question, do you think Hypena need a critical density of pale swallow-wort to maintain their population?
Yeah, I would pass that to any of the primary investigators or researchers that are on the line.
Yeah. Anybody wants to
Yeah,
take that?
yeah. If Robert, I also see Mariana's on the line as well. Any thoughts?
Hello, everybody. Can you hear me?
Yes.
So this is Mariana Such. I'm at Michigan State University, and we have done several common garden experiments where we transplanted plants, so like 40 to 60 stems. And when we released like 5 pairs, they put 5 pairs of adults, so 5 females and 5 males. The resulting larva ate so much that they just devoured those 40 to 60 stems. So that was definitely low stem density for 5 females. But as we know, sometimes you have 5 females and only 2 of them lays eggs. So it's really difficult, I think, to estimate how many larvae you end up with and how much foliage those need. And we were also doing for our releases the approach of confining the adults to lay eggs only for a week or 2 and then removing the cages so both the adults and larvae can move, because you never know when they eat themselves out of food essentially. And we didn't see establishment in those cages where they consumed all the foliage and we couldn't give them more foliage. I mean, in some cages we added extra plants, but it's too little too late.
Yeah, the stripping of the cages is pretty common in terms of, or that's when you're getting establishment and you got to get them open to get them out so they've got more to eat. So they do. The other thing I would say is in terms of the rearing stuff, I'm sure everybody that's reared them, quite often when you look in a cage and think, oh, there's not very much there, I don't see a whole lot of eggs, and then you go back a few days later and boom, there's nothing left. The plants are defoliated. So the larvae are pretty hard to see even when they're very small. And so their ability to strip everything quickly is pretty high. So they definitely can, if they're on a very small patch of low density, they could strip it, but that's not really one of our problems right now. So.
Just one more thing I would add is that those people deciding to confine them until they see defoliation, I wouldn't wait until you see full defoliation, because what we are finding during rearing, that even when we have little foliage left and we think they are still fine, it's actually the larvae is really sensitive to starvation, and then we lose lots of larvae like to starvation when we add foliage too late. So again, I think the cages shouldn't be kept on for longer than 2 or 3 weeks after adults were released. And that's just my personal opinion. And we are also seeing much better performance in the shade than in the sun.
And I would concur with that. 2 years ago, we did some experimental work at Wehle, in the Wehle forest, and we placed one cage in almost full shade and another cage in direct sunlight. And the shade cage very much outperformed the sun cage.
Okay. Yeah, that kind of actually hits home with this next question I was going to ask about does the team have protocols or a gut feeling about what makes a good release site habitat wise? Sun versus shade versus a mix. Minimum. Well, we talked about minimum density of swallow-wort already. If anybody has anything they want to add to, any thoughts on that as far as best selection for release site?
Yeah, it seems the shade sites overall are the best bet from what I've heard from all the research group. And also I think that makes sense because from what I understand, this species was also found in forested sites in its native range in Ukraine.
Oh, here's one from specifically to Audrey, when I think this one actually happened when Audrey was finishing her presentation. It says, Audrey, what was the feedback from partners about the monitoring tools? Both vegetative and insects. Did partners feel confident that they could do the protocol? Did you feel confident with a one-time training, or how exactly did you equip the partners?
Yeah, sure. So for the insect monitoring, we just had that one sheet that I shared earlier, we just went through that sheet briefly with the people who were going to monitor, just like walking through what everything meant exactly, and then people were sent out on their own to do it. And I got the impression that they were pretty confident with that. I think that one was pretty straightforward, just a yes, no, did you see it? Although I will say it is, so this was sort of the test year for, developing these tools. And so everyone involved were sort of trying to get their feedback as far as like, what questions did they have come up when they were monitoring for these insects? And we're trying to develop like outreach materials and supplemental information, like the video guides that I mentioned, to sort of address those questions that automatically come up for these things. So I would say for the insect monitoring, I think people felt pretty confident, but there were some questions that hopefully we'll be able to address in future iterations and with future resources. For the vegetation monitoring, this year we either Carrie or I were out conducting the vegetation monitoring with people who we were partnering with. And so I would say my impression of people's comfort level with doing it depended on if they were sort of management professionals versus if they were the landowners and maybe didn't have a background in working with plants in the field. And so I feel like we might, moving forward, develop different resources or modify the vegetation monitoring form depending on the audience. And so, yeah, I hope that answers your question.
All right, let me see. I'll do one quick one here, and then we'll have to end. Was all the populations in New York pale swallow-wort and not black swallow-wort? Anybody know? Because I know we see pale swallow-wort up here.
Our populations were pale.
Yeah, I think the black is a little farther down in the Hudson, lower Hudson area, but I'm not sure.
Okay. All right. Thank you once again, Carrie, Audrey, Rob, other people that helped answer the questions just now. But for the presentation and answering questions. Resources for the ELOSC can be found on our website at swallow-wort-collaborative.org. The website includes pages on control and management of swallow-wort, an upcoming events page, mapping tools, a section to post academic and practical research work, and a link with instructions on how to join our listserv. If anyone wishes to post articles of interest or publications on your work, please email them to me with permission to post, and I'll take it from there. Future webcasts will occur as presenters and new swallow-wort management information becomes available. We will make announcements about these webcasts on our website, listserv, and the SLELO-PRISM Facebook page. Before I close today's webcast, are there any other comments, questions, discussions from the group?
Thank you, Rob, and And I guess Rob, all the Robs on the line.
Yeah, thank you.
And Mariana also for stepping in and providing some expertise. I just wanted to comment how useful it's been working with all the researchers that have a wealth of knowledge, and it's been really great getting to know everybody. So this is a big cooperative effort, and I look forward to learning more from you guys in the years to come.
Hey, thanks to everyone. That's all we have for today. I wish to thank Audrey, Carrie, and Rob once again for taking the time to share their expertise with us. Please check out the ELOSC website and feel free to share and invite others to participate with this collaborative. Thank you, and have a great afternoon, everyone.
Thanks, Robert.
Thank you.
Thanks, bye.